Hi there. We'll pick up the Chapter 18 material with the third type of hypersensitivity. Type 3 is immune complex mediated. So we still have antibody involvement, but this time the antibodies are creating little networks, little complexes that are the root of the problem. This is something that happens as a part of the disease process in a lot of different autoimmune diseases. What happens is that you make antibodies to antigens that are circulating in your system. And in the case of autoimmune disorders, those are an autoantigen. When they bind, the bound antibody-antigen complexes form these little networks that can become quite large on a molecular scale, and they can get lodged in places like capillary beds. As a response, phagocytes can come in and start to clear them out. But the ones that the phagocytes miss or don't get to right away can activate complement. That can result in damage to the tissues inside your capillary beds and can extend to some of the tissues and organs and eventually cause systemic disorders. Now, localized type of damage can lead to something like pneumonitis happening in the lungs. So if there's antigens or allergens that are being deposited in the lungs, you may get this immune-mediated type of inflammation and damage to the lung tissue. Certain types of jobs predispose people to this condition because they may repeatedly inhale particles with what become allergens. Interestingly, glomerulonephritis, a type of damage caused by a localized reaction, is a side effect of having a Streptococcus pyogenes, also known as group A strep, infection. We'll talk about this organism in more detail in a later chapter. The key point is that if you allow your immune system to clear group A strep on its own, meaning you're not taking antimicrobials to clear it out and your body makes a wide range of antibodies to this organism, one of the side effects that can occur is glomerulonephritis. If you've taken physiology, you might be able to recognize the roots of some of this terminology. Though glomerulus and the nephron are part of the kidney, itis always indicates an inflammation of the specific tissue indicated. So, glomerulonephritis is an inflammation inside the kidneys, particularly happening in the capillary beds, in the glomeruli and the nephrons, where these immune complexes tend to lodge. If you're young and experience this condition, often you'll recover without long-term kidney damage. But as you get older, if you experience this, you have an increased risk of having permanent kidney damage. When type 3 reactions are systemic, that is typically when we're dealing with autoimmune diseases such as lupus or rheumatoid arthritis, where there's degeneration of the tissues being caused by our immune system mediated damage. This is just showing you some signs of these two autoimmune diseases. Rheumatoid arthritis, that is just showing the joints shown up at the top. And we have systemic lupus erythematosus at the bottom with the characteristic rash, sometimes called a butterfly rash, as you see here. In both cases, it's the immune complexes that are depositing in the capillary beds of these tissues that's leading to these physical manifestations. In lupus, this is happening throughout the entire system, but the inflammatory damage is visible right here on the tissues of the face. Okay, so that explains the mechanism of type 3 hypersensitivity, the immune complex mediated reactions. Understand this mechanism, what's happening, and that it can be something that happens as a result of the immune system response to a specific type of infection, like with group A strep, or it could be something that's happening in autoimmune diseases. The last type of hypersensitivity is type 4, or delayed cell type hypersensitivity. This is the only hypersensitivity that is mediated by T cells specifically. We're talking about the cytotoxic T cells. The classic example of this reaction is what's occurring in the tuberculin skin test, where they inject a tiny little bit of the antigens for Mycobacterium, the ones that cause tuberculosis, and then, if you've been exposed, your T cells migrate to this area over the period of a day or so and cause a cytotoxic type of response in the cells surrounding this area. It's called delayed because unlike type 1, which happens very quickly, within minutes, this can take longer, 24 to 48 hours before you see major response. Contact dermatitis is also this type of reaction and is something that you can see with poison ivy or poison oak. These plants have antigens on their surfaces that our T cells recognize and respond strongly to. They get overreactive and you end up with this seeping, weeping, itchy type of response. Now we're going to talk about graft rejection, otherwise known as organ rejection, also a type 4 cell-mediated type of response. Grafts are transplanted organs or tissues, and they can be perceived as foreign by the receiving body. What is happening here is that the MHC on the graft cells is what's being recognized, regardless of whether there's an antigen being presented or not. The MHC itself is interacting with the T cell receptors, and it's recognized as foreign, so therefore the T cells can get very excited over these MHC molecules and bind to them very strongly. The cytotoxic T cells become activated and expand to respond to this foreign tissue. That's going to lead to rejection of the graft tissue. So, the closer you can make MHCs match on individuals, the less likely you're going to see this type of reaction. Ideally, the alleles, the variants of the MHC genes, match perfectly, so you can avoid this response. But that's not typical unless you have an identical twin. This is the concept behind tissue typing for organ transplants. The MHC genes are being examined to try to match them up. The more the genes match, the better the prospect of acceptance of the graft and avoidance of transplant rejection. Some terminology to be familiar with regarding grafts. An autograft is what might happen if you burn yourself and they have to transplant skin from one area of your body to another. So that's a graft or transplant to self. If in the future they're able to use your stem cells to grow you a new organ to transplant, that would be an autograft. Obviously, you would not reject self, so this is an ideal type of transplant. An isograft is a genetically identical transplant. So if you were lucky enough to have an identical twin and they donated an organ to you because yours was failing, you would receive an isograft. This is more common amongst research animals because there are colonies of research animals, such as mice, that are genetically identical to one another. This is useful for researchers trying to figure out what the immune system is doing when they can eliminate the genetic variability. An allograft is what's routinely happening in the organ donation system, where you're getting an organ from the same species, but it's not genetically identical individual. You would be getting the organ donation from another person, even if it's a family member. Lastly, there are xenografts. These don't typically work. This is when you take a tissue from one species and move it to the other. They have tried that, like take the heart of a chimp, for example, and then transplant it into a dying human infant, but it has not worked. The graft is rejected because it's too foreign. They are exploring doing something like humanizing animals. You would grow them up for the specific intent of using organs to transplant. Those animals would have to be genetically modified so that the animal is expressing human genes as opposed to their own species. This is theoretically possible, but it's also likely that researchers will figure out how to grow human tissues in the lab for transplantation, which is more appealing to many people. Medical researchers already have made progress in growing a replacement ear, for example, and are trying to grow liver lobes and lungs. Perhaps someday in your lifetime, this will be a possibility for people needing transplants. They could have a replacement grown from their own cells, as mentioned earlier. The graft versus host disease is a type of rejection, also type 4, but this usually happens when somebody receives a bone marrow transplant. What happens in this case is that the T cells from the donor, who have learned to recognize their previous host as self, will find themselves in the recipient, not recognize it as self, but as foreign and become activated. So the T cells attack the recipient because they have not learned that this new environment is self. So in this case, the graft is actually attacking the new host, and typically this has to do with, again, mismatching of the MHC genes. They can only really treat this condition with immunosuppressive drugs, but that's not ideal because it leads to a person that is very immune compromised and can end up with opportunistic type of infections. They try to improve the success of bone marrow transplants by removing the mature T cells from any potential donor marrow, but it does not always work perfectly. Lastly, I'm going to talk about some other disorders of the immune system, the ones under the category of autoimmune and immune deficiency. There are a wide variety of different autoimmune diseases. I only provided you examples of two, rheumatoid arthritis and systemic lupus erythematosus. There are many others. Most autoimmune disorders are due to improper antibody production to a self-antigen. Although in some of the different disorders, they think a cytotoxic T cell comes in and does damage to neurological tissues, for the most part, a lot of autoimmune disorders are the antibody-mediated type. Some are very specific and mostly one organ or cell type is affected, like red blood cells. But in others, like lupus, many other different organs are affected. This is showing the two examples that we used earlier. In rheumatoid arthritis, the body primarily attacks the joints and causes a degeneration of the joint tissue. Lupus, however, is widespread, although you can see the rash on the face as a characteristic sign of the affliction. Not visible is the same types of damage that is happening in the capillaries of many different tissues of individuals with lupus. One of the interesting things about lupus, as with many autoimmune diseases, is that they tend to be more common in women. Researchers think that perhaps some of that might be related to hormonal makeup, where estrogen may be playing a role in stimulating the cytotoxic T cell. There is also the belief that maternal cells can trigger an autoimmune reaction. So, your mom's cells may be present within your body, and they think that sometimes later in life that can trigger autoimmune disease, and vice versa. Fetal cells have been found in moms as well. This can have the same effect of triggering an autoimmune disorder. They also know that viral infections and sometimes environmental factors play a role in autoimmune reactions. There are genetic factors that predispose you. For example, if you have specific alleles of MHC, they find greater percentages of those individuals with specific autoimmune disorders. Having the MHC allele does not automatically mean you will have the autoimmune disorder, but it increases your chances. The marker is not enough to cause the disease, but that might predispose you, and combined with other things like environmental factors or viral infections, that increases your susceptibility to autoimmune disorders. People, especially women, with specific MHC alleles are more likely to get specific autoimmune diseases. The mechanism is not understood in detail, but the statistical correlation is there, and research in this area is being conducted. They also think that for some of the neurological autoimmune type diseases, cytotoxic T cells come in contact with self-antigens through a traumatic event or through an infection that results in leakiness of the blood-brain barrier, and that can allow self-recognizing lymphocytes to be activated when they previously were not. Previously, these T cells would have been separated from their antigen and didn't get activated. With a leak in the blood-brain barrier, the T cells enter and activate, causing the disorder. So these are some of the factors that seem to be leading to autoimmune disorders, but one in and of itself is not going to be enough. This is an active area of medical research because there is still quite a bit that's not well understood. Why exactly do the protective mechanisms of the immune system break down and disease results? That's the overarching question to be answered. Overall, autoimmune diseases are a result of a failure of normal regulatory control mechanisms that the body has in place for our immune system. When these controls fail, for whatever underlying reason, the autoimmune diseases develop. Now we're going to talk about immunodeficiencies. Immunodeficiency can be grouped under the categories primary or acquired. Primary immunodeficiency means you're born with a condition. You have some genetic mutation or something happened during development and you end up with an immune system that's not functioning properly. There is a wide variety of genes that can be altered that would lead to non-functionality of some aspect of the immune system. The immune system is complicated, and many cells with many active genes must interact. If one is incapable of interacting as it should, then the immune system will have some inability to respond in a typical manner. Acquired immunodeficiency means it happens later in life, and there is some other root cause to it, not genetics. This may be a result of HIV infection, for example. You're exposed to high doses of ionizing radiation that can do some serious damage to your stem cells, which can then lead you to not have sufficient numbers of lymphocytes. This table from your book gives you a flavor of some of the more common immunodeficiency diseases that are directly linked to genetic. But there are quite a few different ones out there. There are a lot of different genetic conditions that can result in an immune system that is not functioning properly. You don't have to memorize these. While in the past it might have meant that the individual would likely die in early childhood, that's not necessarily true anymore because we do have therapies that can help a person live a fairly normal life. For example, passive protection is employed if somebody cannot make their own antibodies, like in Bruton type A gamma globulinemia. In this disorder, the person does not make their own antibodies, but can be provided pooled antiserum from the population. Blood donations are separated for the liquid portion and the antibody fraction purified for administration. People with such conditions will, on a routine basis, go in for an infusion so they can have a normal antibody concentration circulating in their blood that can help clear invaders. Since antibody degrades, the pooled antiserum must be re-administered regularly. There's a lot of interest, too, in correcting the genetic defects that cause these diseases. For example, by taking stem cells and correcting the mutation and reintroducing the modified cells to the individual. This is gene therapy. That treatment possibility is probably coming in your lifetime because of some of the better genetic tools researchers have available today, like CRISPR-Cas9. This system is one we have not talked about, but it's a big deal, and it's going to improve the ability to genetically manipulate cells and then return them so that they can function properly. Older methods of gene therapy have occasionally been employed successfully, but CRISPR-Cas9 systems hold even greater promise to alter, to correct genetic variants that are detrimental to the individual. Next is acquired immunodeficiencies. These happen not as a result of genetics, but as a result of something that happens later to the individual. Some acquired immunodeficiencies could result from severe stress. We can see evidence of this during finals week. During finals, if you look around at your classmates, you find many of them suffering from colds and other infections. When you are under extreme stress, especially when you're not eating and sleeping properly, you will suppress your immune system. This is well documented. When people are stressed out, they have more susceptibility to infections. Also, think about the disease called AIDS. This is where we have HIV virus infecting CD4 positive cells and causing them to die and be eliminated by your immune system. Those CD4 positive T cells are very important for the functioning of your immune system. So when those and the concentration of those cells in your system goes down, you experience immunodeficiency and are very susceptible to infections, even from organisms that normally would not cause a healthy individual to be infected. We have already talked about HIV and its infection cycle, but in this context, we just want to mention that AIDS is the result of infection with HIV, and it's not a single condition. It's a syndrome, meaning there are several things going on simultaneously. One is, that's important of course, that you have to be infected with the virus, and the virus gets inserted into the genome of the cells of the individual. If you remember, macrophages and monocytes also serve as reservoirs and can spread the infection to the body because they don't effectively clear it out. This is a close-up of the HIV, a cartoon version of the virus. The main players, the capsid reverse transcriptase, which turns RNA to DNA, integrase, the enzyme that integrates the viral DNA into the host DNA, protease being important for the maturation of the virus. We have attachment proteins, here GP20 and GP41. We have the genome, which is RNA packaged inside a nucleocapsid inside the envelope. Look over this slide and make sure you're able to explain what's going on. You can use that HHMI life cycle to review the important enzymes and the steps of viral replication. But what I really want you to focus on is this slide in the different infections that are opportunistic that will occur in individuals that are slipping into the condition called AIDS. Remember, AIDS is the disease caused by HIV. Once the CD4 positive T cell population takes this big dive, the person is very susceptible to infections that would normally not cause a problem. One that I want to draw your attention to is this specific pneumonia called pneumocystis, right here. This infection by this fungus was really one of the key alerts, a red flag, that is something unusual was happening when HIV first came onto the scene in the 80s. Doctors saw this type of pneumonia occurring when they never had seen it before, and they realized it was specifically linked to HIV-infected individuals. That was a clue for them to realize that the immune system of these individuals was not functioning properly. These individuals were susceptible to normally harmless microorganisms. And just like pneumocystis, a lot of these different infections do not appear commonly in the normal population, but are very common in people that are suffering from AIDS. These are opportunistic infections. We covered that concept earlier. This slide has a lot going on, but it's tying together the whole picture of what's happening during HIV infection and the development of AIDS. Let's first look at this red line. That's the concentration of virus in the blood. Early on, it spikes, and then as your immune system sort of learns how to respond to it, the numbers come down, but it can't eliminate the virus. The virus is sneaky, and it's hanging out in your immune cells, and it's integrated into the genome. It's also mutating surface proteins to avoid elimination with antibodies. So the virus keeps getting made, and our immune system keeps trying to eliminate infected cells. Over a long course of time, notice we put a break in this axis line because it could be years. Your immune system gets tired. It can't keep up, can't replace all its cells. When the immune system loses the ability to eliminate and replace the infected cells, the virus concentration will start to rise and then sharply increase. Once you get that big peak in viral numbers, that is really a signal that your immune system is not able to effectively deal with it. If we look at the CD4 positive count in the green line, the cell type that HIV infects, it's our immune coordinator cell. Here, as the virus numbers are peaking, the CD4 numbers are crashing. That is bad for the individual. If we look at the antibody response, now in blue, remember the CD4 positive T cells are needed to stimulate these cells to make antibodies. So yes, the individual makes antibody and that concentration goes up, but remember that is not a protective antibody. It doesn't neutralize and clear the virus from the system. That antibody concentration is going to go down later. Why? Because as you're killing off the CD4 positive T cells, you're not able to stimulate the B cells to make antibodies anymore. And so that concentration crashes as well. So how do we treat it? I'm going to leave you with this thought. Remember we talked about when we use antimicrobial therapy, it really should be a cocktail to protect against resistance. The different antimicrobials should have different targets to work best. In this case, the antivirals must have different viral targets to really be effective. The drug should affect different viral components. If you just have one target, like reverse transcriptase enzyme, the virus will become resistant to that drug fairly quickly. Mutations in one gene can confer resistance. So the goal, And the key to delay the progression to AIDS is for antiretroviral therapy to keep the viral replication and the viral load low and suppressed. For the therapy to do this, it needs to include a cocktail of drugs with different targets in order to really reduce the replication and hopefully keep the individual healthy for a very long period of time. It's very unlikely that a viral mutant will occur with the right mutations in more than one gene at the same time to resist the effects of the therapy. This is why the cocktail is so much more desirable than using a single drug. We have not had a vaccine developed yet. This is because of the mutation rate of HIV. Researchers have been searching for and have not yet been successful in locating a target for antibodies to bind that can neutralize all the different variants of HIV. If one can be found, it will obviously be a game changer for dealing with this terrible infection. All right, that's it for Chapter 18 material. I'll see you in the next one.